welcome everyone to UC Irvine's School of Medicine and STEM organized session to learn what goes on behind the scientific publishing. And today's speaker is Dr. Ruth Zearfoss. I would like to first thank Professor Tali Baram from School of Medicine. Without her initiative, this webinar wouldn't have been possible, and she actually went out of the way to invite Dr. Zearfoss to UC Irvine. So at this point, I would like to invite Professor Tali Baram to introduce our speaker for today and kick off the event. Thank you, Dr. Baram.
Thank you, Harinder. Again, you made the whole thing happen. And it really is a pleasure because I think that publishing is, it's clear as bread and butter, as you said, is something we need to know about. And I have been very impressed with the fact that many of the scientific editors that we deal with are really scientists and made the decision to move and become editors. But very few that I've been familiar with actually take the trouble to explain this to us and seem to be passionate and interested in sharing what really goes on. And when we interacted with Dr. Zearfoss in terms of our publication in Cell Report, it became evident that Dr. Zearfoss has done it before. So I was really thrilled to ask, would you be willing to come? And the answer was yes. So Dr. Zearfoss, as is written here now, is editor-in-chief of Cell Report Method, which is just being launched and has worked with a variety of roles in cell reports. She got a scientific education initially in Houston, so did I, and then moved on to University of Massachusetts and spent some time there. And in 2004, realized that perhaps a better career choice would be in editorial realm and publishing. And we are fortunate for this decision because we'll learn a lot today. So thank you again for coming.
Thank you so much. Thank you, Tali and Harinder, for putting this together. I'm really excited to be here. I love speaking to everyone about what goes on with their papers after they submit them to a journal. I really hope that this is an interactive conversation over Zoom as much as we can. So please write down your questions and send them in. We can also discuss career path things once I've finished the behind the scenes part of the presentation. So I just invite you to, whatever your question is, please ask it. Whoops, wait a minute. I can't move my screen. Okay, so I hope everyone can see this now. So I'm just going to give you an overview about what I'm going to talk about today. So just the basics of putting your paper together from the perspective of an editor. So what is it that we're going to look for when we see this paper? What happens to the paper after you send it into the journal? I know when I was a graduate student and a postdoc, it seemed like I pushed a button and something scary happened and I just had to wait. So it's really a lot does go into what happens after you push the button. So I'd like to kind of share whatever I can about that. How we handle the reviews, what we look for in the reviews, how we decide to give weight to the different reviews that we get, how we communicate the decision to the authors. And then just some best practices, both for authors and reviewers, because authors and reviewers are the same people. And I think it's easy to lose sight of that. And so you want to keep that in mind at all times. And then I'll just give you a little bit of, I guess, an advertisement for Cell Press, Cell Reports. And the new journal has not yet been launched, but it will soon be Cell Reports Methods. So basically, a journal is fundamentally a collaboration between a lot of different people. It takes a lot of work to put a journal together. Primarily the authors who they conduct the experiments, them submit the work. That is the core of the journal. The editors, our job is to collate a cohesive set of work at a certain quality level, looking at broadly at the content of the paper. Scientific topic for what we've designed as the scope of the journal that we're putting together. The reviewers assure the scientific quality of the work, and they also give us some broad insight into whether they think the work is interesting. And also our editorial board, they're an invaluable source of information. We reach out to them for advice a lot. And we all have the same overall goal. So authors, editors, reviewers, and our editorial board, we all want to publish papers. We all want to publish good papers and put them together in a way that's going to advance the knowledge of the community in a reproducible and solid way. And that's the goal. So the first step is writing and submitting the paper. And I know for me, it seemed like all of the work was in this first bullet point here, doing the research before writing. But really, it's worthwhile to pay attention to the steps that come beyond that, such as choosing a journal. There are lots and lots of journals to choose from. And they each, a lot of them have a unique scope that they're looking for. So you want to make sure that the paper that the journal that you're choosing is broadly similar to the type of paper that you're trying to submit to the journal. Preparing the manuscript and preparing the cover letter. I'm going to go through each of these in more detail here. So the first is building the manuscript, putting the manuscript together. It's when editors are reading papers at a busy journal, we have a lot of papers to read. And one of the most useful things you can do is in a clear way at the beginning of a paper in the abstract, hopefully, you want to identify the gap in knowledge that you're filling. I would say that's probably the most important thing that you can do when you're putting your paper together. You want to make that clear. You want to know, the editor wants to know why you're asking the question that you're asking. To do this, you want to cite the current literature, because we do go through PubMed and see what's known in the area. So you want to cite the current literature to put your paper in the context, explain how your paper is building on what's currently known, or if there's conflicting data in the field, you want to explain why your finding is going to fill this gap that's present because of this conflicting data. And you do want to make it simple and concise. I think a lot of times there's a tendency to make things seem complicated, and really the simpler things can be, that more easily they can be communicated to a wide variety of people. You want to plan your figures in a very organized way that'll lead to a logically structured argument. And it's also a really good idea to ask people outside the field for feedback. A lot of times if you're working in a field and you live and breathe this field all the time, there are questions, things that would seem obvious to you that wouldn't be obvious to someone who doesn't work exactly in that field. And if you're submitting to a broad journal, you're going to have readers from a wide variety of fields. So you want to ask somebody outside the field if they understand what you're doing, if they understand the argument that you're presenting. And as I already indicated, I think more is not necessarily better. So you do want to keep it concise and get to the point. And if language assistance is needed, I do recommend that you seek this. We try really hard not to let a language barrier impact our assessment of a paper. But sometimes if we can't understand something because of a language barrier, it makes things harder for the authors. So I would recommend that. So choosing a journal. Does the paper broadly fit within a journal's scope? So for example, at Cell Press, there are a number of different journals you could choose from, and we each have a defined scope. So for example, Molecular Cell might be looking for a very solid novel mechanistic insight into how something is happening, whereas Developmental Cell is looking for more of a biological advance into how a part of the organism is developing. So you want to make sure you've targeted the advance that you see in your work to the advance that the journal is looking for. Does the journal publish broadly similar papers? So we recently started publishing reviews at Cell Reports, but I can tell you, we always had people submit reviews to us when we didn't publish any of them. So I think that you can save yourself some time just making sure that the journal is requesting papers that are in line with what you're sending them. You also want to think about the audience that the journal has. If you want people in a certain field to read your paper, you might target that paper for a journal that has a lot of readership in a certain field. Do you read that journal? If you don't, then maybe you might not want to go there, or maybe you would if you want to break into a field. If you're unsure, you can consider sending a pre-submission inquiry. If anyone has questions about those, whether they should do it, I'm happy to answer questions about that later. We evaluate those very quickly, but you can get a broad sense of whether a paper might be in a journal's scope by doing that. And then beware of impact factor obsession. I think you probably will make your life easier if you judge based on the quality of the papers in a specific field that a journal publishes, rather than sort of like starting at the top and going down. So once you decide to submit your paper to a journal, you'll have to write a cover letter. So this is a really useful place to communicate anything with the editor of the paper that you need to say that you couldn't put in the paper itself. So you want to summarize basically the paper's broad relevance and scientific advance in a concise way so that it's clear, and lay this out. This is also a place where you can let the editor know anything else that might be important and relevant to the papers. For example, reviewer exclusions. At Cell Press, we allow 3 reviewer exclusions. People can be excluded for any reason because we understand that it might be necessary in some cases. And you don't have to justify why you're excluding someone. And we don't go to those people. So reviewer suggestions, I love reviewer suggestions because I find them helpful. Sometimes authors will suggest reviewers who are the perfect people. Sometimes they suggest people who are not so appropriate. So don't put your postdoc advisor as your suggested reviewer because we do look for conflicts of interest there. But it can be really helpful for us to see who was suggested to review a paper. And then another thing to keep in mind, if there's anything else related to the paper that we might need to know. So for example, if there's related work in submission elsewhere, if there's an urgent competitive situation, anything else that you need to communicate, that's really what the cover letter is for. The cover letters are confidential. They're not going to be shared with reviewers. So if there's a situation that is important, this is the place to mention them. So that's submitting the paper. So what happens after that? The editors will assess the paper. And so what do we look for? So hopefully I can summarize this. So I think every journal at Cell Press, I think, may have a slightly different way of doing this. So I'm going to tell you the way Cell Reports does this. So each paper gets read by an editor thoroughly. There's a single editor who handles each paper and reads it in depth, but we do discuss all of the papers as a group. So we have input from multiple people on every paper. Occasionally we'll seek external advice on a paper. So there's some papers that are faster and some are slower to assess. What we're looking for is the core scientific advance that is presented in the paper. We also try to get a sense of the approaches that the authors are taking. So are the methods that they're using broadly applicable to the question that they're asking? And are they up to date, or are they perhaps not appropriate? So after doing this, we'll make a decision whether to send the paper for review. If we do send it out, we select 2 to 3 reviewers who are people in the field to review the paper. And if we don't send it out, we try to explain as best as we can to the authors why we're not sending it out. So what are we looking for? The first is just a simple scope issue. Is this paper in the scope of the papers that we're trying to publish? Does it address an important question? So sometimes that can get a little bit subjective. A question may be important to one person and not to another person. So that's what editorial discussion is for. And that's also where we might reach out to someone in the field to get insight into whether this question is important or not. How much do the findings advance the field above what's already known? So we do PubMed searches for all of these, for every paper that we get, to try to see, how big is the increment of advance that's being presented in the paper. So for example, if a protein has 4 phosphorylation sites and you found one more that does the same thing as all the other ones, we might, we see the advance, but we might say that's probably a more incremental advance. It's probably better for a field-specific journal. But a whole new branch of a pathway leading to a new function, that would be a more significant advance. So we're trying to tease out these issues as far as the impact that this finding is going to make. For Cell Reports, we're a broad journal. So we also ask ourselves, are these findings going to be of interest to scientists outside the immediate field, or are these findings going to be of interest only to the people who work exactly on that process? And are the experiments broadly appropriate to answer the question that's being asked? So we send the paper out, we get the reviews back, and what do we do? So this is, I actually have to say, I love reading reviews. So we read the reviews very carefully. I've actually had people ask me, do you read the reviews? Yeah, we do. We read them very carefully. It's very common that reviewers disagree with each other. It's very common. And we have to somehow resolve that very frequently. So I have a paper right now. I think I have one review for accept and one review don't ever publish. So we have to sort this out. It's not simple vote counting. So every paper has multidisciplinary aspects to it. You know, there might, so we end up trying to get reviewers to cover all of these different areas. So we'll have, some reviewers might be a technical reviewer who are brought on for us to evaluate a specific technique, if that's an unusual technique that requires some specific training. Some reviewers might be for a more biological perspective. So a lot of times we look to different reviewers for different things. And if the technical expert isn't happy with the paper because there's an issue with that, that could be a fatal flaw. Or if the technical expert has a conceptual issue with, just doesn't think it's interesting, that might not be what we're going to that reviewer for in the first place. So it really becomes an assessment of the overall decision based upon who the reviewers are and what we were asking of them. We always discuss the reviews with each other also before we send a decision to make sure that we're sending the right decision for the paper. And then we send the authors a letter to let them know what the decision is based on the reviews. So what do we ask of the reviewers? The first thing is the reviewers, the core function for us is that they are there to evaluate the technical quality of the data. Are the controls present? Are the experiments conducted right? Did they normalize correctly? Do they have their p-values and statistics done appropriately? We don't assess those things before the manuscript goes to review. We recognize that papers can always be improved through the review process. And to a lot of times, those things can be improved and make a paper that might have a few things missing could turn out to be a really great paper. Reviewers are also asked to evaluate the degree to which the data support the conclusions. If there's really no support for the conclusions that the authors are drawing, that can be a fatal flaw for a paper. So we look to reviewers for that. And also provide feedback on the level of interest to those, either within the field or more broadly, depending upon what our questions were about the paper. And how do we come to a decision? So I already indicated reviewer expertise. That's really important. The feasibility of the suggested experiments. There can be times when an author may be asked to make a conditional knockout, do single cell sequencing, some very hard experiments that might not be within their capabilities. So we try to take that into consideration when we're making a decision for a paper. We generally would like revisions to be done within 2 or 3 months for a reasonable revision. So we also look to the reviewers for conceptual and general interest points. I think I've already said that. And occasionally, in cases where we might have a split opinion, we can consult with the reviewers a little bit more. So sometimes we'll go back to a reviewer and we'll say, hey, are you sure about this? You didn't like this paper, but we have 2 reviewers that really like it. What do you really think? So we do this fairly frequently. Also, our editorial opinion. Do we think there's a lot of interest in this area based upon what we see? And so then we communicate this to the authors. So we try to focus on the scientific issues, what will help improve the study. I think that's the most important part. When you're reading these letters, I think sometimes people might expect there to be a secret code, but there's not a secret code. Does the editor explicitly invite a revision? That should be clearly outlined in the letter. And if it's not, reach out to the editor and we should clarify that. Do we mention resubmission? Do we mention any specific points that we think are important to address? If you disagree with the suggestions or the decision, we are always here to discuss these things. We tend to get very busy, but we are here and available and willing to discuss these things if you reach out. I know it can be sort of an emotionally charged time if a decision if you feel a decision is unfair. That's why it's really good to focus on the scientific points and avoid any emotional points because they tend to be not productive. So for a revision, what you want to do is address the criticisms that the reviewers have raised as well as you can and as thoroughly as you can. That's the best path forward. Prepare a point-by-point response and resubmit the paper after doing the experiments. So a paper may have been declined. And if this is the case, if you have a scientifically based reason that you think that this is not the correct decision for the paper, you can submit an appeal. So we don't just discard all appeals. We do discuss all of our appeals. We recognize that mistakes can be made, and there can be cases where we send a decision to an author expecting them to appeal, and being very open to an appeal when we send that decision based upon the comments that we have from the reviewers. Because we do look to reviewers for technical points to be addressed. They're the technical experts. So it may be that we're rooting for a paper, but we still may have to reject the paper, but we may be very open to reconsidering it if it's a scientifically based argument. So if we did not invite a resubmission, the paper was declined. If there are a lot of scientific valid criticisms, it's probably in your best interest to go elsewhere. If you do choose to appeal, a new data is probably the best path for success. If you can provide data, numbers, analyses, statistics to show that your claims are correct, that's going to be more effective than arguing. And be realistic. I think if you're not realistic, you can waste your own time. That's not to anyone's advantage. Yeah. So final tips. So editors, we do really want to help. We're very busy. We handle a lot of papers. And so that's why I would emphasize being focused on the scientific points and being very concise in the language. It'll make things easier for everyone. And also wait at least 24 hours before sending in a response. We understand people may have an emotional reaction when they get a decision that they're not happy with, but if you wait 24 hours, it can make the conversation go a little bit more smoothly after that. I'm just going to check the time. Okay. So author responsibilities. So when you're submitting a paper, there are a few things that are really important to keep in mind that can kind of trip up the process if you don't pay attention to them. So ethical approvals for animals and subjects is very important. Cell Press now has the STAR methods section. It has a specific section for this. It's really important that this is all approved and done correctly. Any conflicts of interest really do need to be disclosed up front. Any authorship issues should also be dealt with. This is the author's responsibility to iron out all authorship issues. So this includes who is an author, the order of the authors. It's really important this is all ironed out ahead of time. Issues that come up after publication are impossible to deal with. You want to pay attention to your figure preparation. It's surprisingly common to see things like figure placeholders put in. So the wrong gel is included just for spacing purposes, and then somebody forgets to take it out and put the right gel in. So take a special care there, because this can lead to issues about reproducibility downstream. And another thing to keep in mind is we see a lot of gels that have been spliced with different lanes from different gels put together to make it look like it's one blot for figure preparation purposes. And that's really not appropriate. We have to have authors change all of these. So when you're running your gels, make sure that you've got the lanes that you want next to each other at the right time. If you don't have that, just make it very clear in the figure that that's the case. So we want a white space or a solid black line between all the lanes that were not run next to each other on the same gel. Carefully detail experimental design and be very careful about the number of replicates that have been done. You want to be clear about that. All the statistical tests that have been done, be very clear about that. Make sure all the authors agree with the manuscript before you submit it. We do occasionally see, or we have seen, authors that didn't know that their name was on a paper. And you want to make sure your publication is original. Don't duplicate sections of text. And just continuing in that, for publication, you only want to submit to one journal at a time. If there's another related paper under consideration elsewhere, you should let the editor know. There can be times when the data in that other paper would be relevant to this paper, to the paper that you just submitted. And the reviewers might need to review that data at the same time in order to make a determination about the paper that was submitted to another journal. It can get really complicated when that happens. But if that happens, we do need to have access to that other data so that the reviewers can evaluate the current paper fairly. Any conflicts of interest should be declared. Data needs to all be deposited into repositories. And it's really important to provide reagents and raw data post-publication. So reviewers also have responsibility. And the number one is confidentiality. So we do allow collaborators or colleagues to help with reviews, but you really need to run this by the editor before you bring somebody else on board. So junior scientists working in your lab, it's common for them to help in the review, and that's almost always usually fine, but you still need to ask and make sure. It gets a little trickier if it's someone down the hall who's an expert on a specific topic, because we have the conflict of interest information and we can tell whether someone has been excluded. So you really do need to check that. If you're asked to review a paper and you have a conflict of interest that we're not aware of, you do need to disclose this, let the editor know, and we'll tell you if we think that's a serious conflict of interest or not. There's some fields that are very small, everyone knows each other. Those fields can be tricky. There can be relationships, trainer relationships that we would like to avoid. Also, as a reviewer, you want to speak to the author in a way that you would like to be spoken to if it were your paper. So just be polite, fair, constructive, timely. You want to focus on the scientific technical issues and be clear about it, not overly wordy. And you want to be helpful, offer helpful feedback for the authors. You don't necessarily have to be completely prescriptive, but give them an idea of what you're looking for rather than just issuing broad criticisms. You can ask to see the raw data. If you have questions about it, I do recommend that you do ask to see the raw data, because when the paper comes back, that would be the chance to take a look at the raw data and make sure it looks okay. And there's usually a place to send confidential comments to the editor, which is fine. But the tone and content really should match the tone and content of the comments to the authors. One thing that can happen is there can be some very harsh negative comments made to the editor, and the comments to the author can be very positive. And that causes a lot of problems downstream in the decision-making process. And it becomes difficult to communicate to the authors what the concern was about their paper when comments are made confidentially to us that differ dramatically from the comments to the author. So I'm just gonna switch gear a little bit. I'm gonna talk just a little bit about cell reports, tell you a little bit about the journal, and then we can move into questions, and you can cut me off and we can move straight into questions if you want at any point. So Cell Reports was launched in January 2012. So I can't believe it, that was 8 years ago. We're a high quality open access journal from Cell Press. We're very broad. We cover the entire life sciences. We really like the short single point reports. So these are 4 or 5 figure papers that are designed to address a single question. So what we're not looking for in those is a long, big story with the initial finding, the mechanism, the physiological relevance. We're looking for like one component of that. So the goal of that is to help speed the publication process for authors. We do also publish research articles, and we do publish resources. So a resource would be something like a large data set. It could also be a method, although now we're launching cell reports methods, which is going to overlap that slightly. So what makes cell reports unique? I mentioned our focus on reports. We have a very active and engaged editorial board of young up-and-coming scientists, although now some of them have been there for 8 years, so some of them may not be up-and-coming anymore. They're a really big help to us to help us solve any disagreements that we have between reviewers and to help us come to decisions about papers when we need them. We also have the editorial expertise of Cell Press around, although not with the pandemic, but when we were in the office, we did. Everybody's on Zoom now. They give us advice and facilitate the transfer process of papers, which can help speed the process for authors. So what does open access mean? Readers have immediate, unrestricted access to all papers published in Cell Reports. Authors retain the copyright. We have 2 different licenses. I'm not gonna go through these 2 licenses in detail. You can look them up if you want to. They're basically, one is more restrictive, one is less restrictive. The more restrictive one, I think users are not allowed to modify it or distribute it, and it cannot be used commercially. So the benefits of cell reports, the transfer system helps authors publish more quickly. They can use the reviews from one journal, transfer the paper to Cell Reports, and we'll consider it as a revised paper and not as a new submission, unless the author requests us to consider it as a new submission. We try to have a turnaround time on initial decisions in 3 to 5 days. We're publishing now on a weekly basis, and we have an online format, including graphical abstracts, highlights that convey the message in a very succinct, fast way. So just to give you an idea of the numbers of papers that we review and publish. Most Cell Press journals send, I would say, 40 to 50% of submissions for review. The initial decision on that is usually made in about 5 days. Reviewing usually takes 3 to 4 weeks. And I would say, broadly speaking, Cell Press journals publish 10 to 20% of new submissions. We ask for only 1 major round of revision. There might be a case when we allow authors to go through another round of revision. We try to make that minor and not ask for much at the second round of revision. So I've just put our current editorial board here so you can see everyone. Cell Reports has grown a lot recently. So I just want to thank all of these people. Everyone is very hardworking, and they've made working at Cell Reports a joy. They do a great job. And then I'm just going to spend just a minute to talk about Cell Reports methods just being launched. We're hopefully going to have our EM, which is Editorial Manager Submission System, active, I hope in December, and a website active, hopefully in January. We're going to focus on methodological and technological advances. So what that means is that if you have a paper that has a new method, and it doesn't take what we always say at Cell Reports is that next step, is where you use the method to reveal a biological finding. If you don't have that next step, Cell Reports turns away a lot of papers for that reason. Cell Reports methods is going to be a place for those papers. So we're not going to look for a biological insight in these papers. We're going to look for a methodological advance. So something that people are going to take and use to address their question. We're going to focus on reproducibility, open access publication, just like Cell Reports. We're going to have the short single point reports as well as longer, more complicated methods. And like I said before, we're not going to ask for that next step of the biological insight. So our advance is going to be in the method itself. And that's all I had. I think I've left a lot of time for questions here. So I really am happy to take any questions anyone might have. And there's not anything you should be afraid to ask, because I've been a graduate student and a postdoc, and I know it seems like a mystery. So
Thank you very much, Zearfoss. This was very, very useful. I think a lot of things got covered during your presentation, but still there are some questions which are very unique and I think very interesting ones. I see that there's one question in the Q&A box. So I have a whole category for that box, and we will for sure get to it. There are a couple more, based on that. If you have more questions, add them into the Q&A box. But what I'm going to do now is I'm going to go to the questions which a lot of you have submitted when you were registering. So let's start out by asking one question. There's an interesting question about, reviewers. Like, how are the reviewers selected?
how are they selected? Okay, so we basically try to make sure every scientific area that we need covered is covered. Now, this is Cell Reports. I think different journals have slightly different ways of finding reviewers. And I'm not talking about like how do we search the internet for them or how do we search systems for them. I'm talking about like conceptually how we find them. And so if you need to revise the question, please feel free. But so let's say a sample, a paper is about limb development and they do single cell sequencing, for example. I need to make sure I have a single cell sequencing expert on it. I need to make sure I have a limb development person on it. Those are the two fundamental things. I would also hope I would have somebody, one of those people or somebody else who would give me a broader insight into whether this question is of interest in general to developmental biologists.
Great.
Before you go to the next question, I'd simply like to add my thanks. This is a total delight. I mean, logical and systematic, and exactly what we need to do and exactly what we need to know. So this is a lot of fun. Thank you.
Another question, which is again based on more review process. So the person wants to know, what's the common reason a paper gets rejected by the journal, not like before it even reaches the reviewer? I know you did mention some of the aspects, but something which is very glaring the moment you look at it is like, okay, this is not going to make it.
Okay, that's a really tough question because there are a lot of different types of papers. Okay, the most common reason a paper would be rejected editorially. It's very common that we don't think a paper is a conceptual advance. So a lot of times when we go to look in PubMed for related papers, we find a lot of overlapping papers. So we might be able to pull up 50 papers on the same broad topic. And a lot of times we can see that there's a new aspect to the story in the paper that we're looking at, but we think that the overall interest in the field isn't enough to drive interest in that particular finding, given the background of information that's already out there on this topic, if that makes sense to you. So there might be cases where something that looks nitty gritty would be of interest. And that might be if it's a longstanding question for some reason, if it's something that causes a dramatic biological, physiological change, if something's on or off, if there's a biological context where this is required, or if it's been a longstanding question, or if there's disagreement about this. So there might be cases where something that looks like a busy field and a small advance might be more of interest. But that's, I would say, the main reason things are rejected editorially. Yeah. And so we try really hard to sort that out, and it can be trickier. And if we feel it's tricky, that's where we try to get input from the field.
Okay. We have one question from Jessica. I think, there was something very similar in the questions people asked. So I'm going to combine those two questions together. So this person wants to know, is it important to have latest and cutting edge techniques for a paper to be accepted in Cell, press journal? And the other person wanted to ask, like, what is more important? If a paper is full of like really cutting edge and latest technology compared to an idea which is very novel, but then people have not used that cutting edge and latest technologies, what would you prefer most?
Okay, so it's not a either or, I would say. So the first thing that we look for is the idea, the finding, the core finding. If the core finding is interesting and novel, we might not expect I'm just speaking for cell reports because every journal is going to be distinct. At cell reports, we love those papers. We want to give those papers a shot. So if there's a very exciting new idea that's different, the methods are not cutting edge, it needs to be explored further to get at the mechanism, we're interested in that paper. There can be papers where it's a finding that was known 20 years ago and you apply the newest method to it and you spend a lot of money, do really elegant work, but in the end you've confirmed what we already thought. Those papers are challenging because a lot of times we appreciate sort of the beauty of what's been done, but we may have to turn that paper away anyway, because if it's commonly accepted in the field, a lot of times that's what reviewers tell us for those papers is everybody already knows this. And it's for us editorially, that's not, we are trying to publish advances, things that will be interesting to people. And if everybody already knows it, it might not be interesting to readers.
Move on to the next question, which is an interesting one. So when you're trying to publish in Cell or a high impact factor journal, there's a myth that maybe you need to have a lot of data. It should be a data packed publication. So the person wants to know, is that reality, or even if it's a shorter publication but very impactful data, that's what you prefer? So
I can only speak for Cell Reports on this. I can tell you Cell Reports, that's the reason we have the report format. We're not looking for a data packed publication as a report. If you submit it as a research article, we're going to expect more. We're going to expect a little bit more physiological insight or a little bit more mechanism. We wouldn't expect it to go as far as a cell paper or as deep mechanistically as a molecular cell paper, but we would expect it to be more in depth. But as a report, that's the reason that we have the reports, is for an interesting finding that may not have so much data about it yet. But we do want it to be a robustly supported finding.
Mm-hmm. Okay.
So
there's another question, which is a little tricky one, and I'm sure we've either experienced this thing or we believe that maybe that's what's going on. So maybe if you can shed some light on, it's an interesting one. So the person says, could you talk about when the peer review process fails? Example, the reviewers delaying a review of a paper to push their own paper.
Yeah, can I talk about that? So obviously we hope this doesn't happen. We really hope this doesn't happen, and we try not to let this happen. I don't know. I mean, I can tell you if it's proven that this happens, if this is proven, this is a hard thing to prove. There are a lot of reasons somebody might be delayed. Well, first of all, the reviewer should not have agreed to review the paper if they have an overlapping paper. So that's a mistake. If it's clear that during that review process, their paper was published while they were holding up another paper, there's a process in place at Cell Press to flag the reviewer so that they're not used again. As editors, we're sympathetic to that situation.
Okay. There were two questions which are very similar, so I'm trying to put them together. Number 6. Okay. So the question is, how do you best respond to a reviewer if you don't agree some of the substantial comments? And the other related one is, it's a nice way of asking a question. Will the editor look badly upon me if I chastise a reviewer for asking me to do work which would not improve the paper?
We wouldn't look badly upon anyone. Okay, wait. Okay, so I'll focus on the second one. The second question. Okay, so if the work that's being requested will not improve the paper. So I would say that work that was requested might not improve the paper from the author's point of view, but it would improve it from the reviewer's point of view. And it might also improve it from the point of view of a broad readership, which we do have a broad readership. So this is fairly common. As an editor, I recommend to focus on the scientific points. There can be times where it's just easiest just to go ahead and do the experiment, even if you may disagree with it, and put it in the paper. And if it's not a very time consuming or expensive experiment, I do think that that's the right approach. There can be cases where a reviewer might be wrong. And if that's the case, I think it's fine to politely say that and say, we think that there's a misunderstanding. We think that there may be an inaccuracy there. The reason is, and then use data to back that up. So you want to avoid making any sort of personal statements. Focus on the data, facts, things that you can present a figure and show something to back up your claim. Cite another paper. Approach it this way. Does that answer both of the questions?
Yes, yes, they
Okay.
do.
Harinder, can I ask a question? Or
Yes, please. No, please.
Do you have many others waiting? Because I
have a couple. Please go. But we do have 10 minutes, so yeah.
Okay. So last week on Twitter, there was an announcement by Zearfoss that you are actually changing the review process and allowing maybe editors to make decisions rather than authors, if I got that right, about which journal is appropriate for a given paper. And several other really, really interesting and potentially transformative approaches to the review process.
Yeah. So
now we invited you to give a talk before last week, but it is October 21, and I'm really wondering if you can sort of illuminate all of us a little bit about that.
Sure, I can talk about that process and the motivations behind it and what we're hoping for. It's a pilot. We don't know how it's going to go, so we're just trying it to try to see if it makes author experiences better. That's the overall goal. So one thing that can happen to papers is they can be bounced around from journal to journal, and it seems to us like this must not be very fun for authors. So the goal is to avoid that. And the goal is not to have editors make the choice. The goal is to have authors make a choice about how they want to revise their paper, if that makes sense. So there's something called the community review. That's the name of the pilot. You would submit your paper to the community, the Cell Press community, and not to a specific journal. And a group of editors who have insight into all the different journals, what the different journals are looking for specifically in terms of their scope, would then evaluate the manuscripts. I think when you submit the paper, I believe, I haven't stepped through it myself because it's new, I believe that you're allowed to indicate which journals you're interested in. So you could pick all the journals, you could pick 2 journals, you could pick Cell and Developmental Cell, or Cell and Cancer Cell. Or you could pick the entire suite of journals. And so then you would get insight into which editors at which journals would send your paper out if it came specifically to their journal. I think then you're given a choice. Do you want this journal to review your paper? Your paper will have one review. You'll get the reviewer comments. And the goal is to then have those comments sort of interpreted in a way so that you can choose then which journal you're revising for. So for example, if you're at Cancer Cell, you might have to do a lot of samples on patient-derived tissue samples. You're going to have really strong therapeutic relevance. But at Cell Reports, we might not need that. If it's a paper, if you chose Cell Reports and Cell Reports methods, to go to Cell Reports, you would want the method itself to be robust, but you would also have to do an experiment where you show that then we use this method to show that X, Y, and Z happen in this tissue. And at Cell Reports methods, we say, well, if you don't want to do that experiment, come to us, we'll consider the paper without that other step. So the goal is to provide authors with sort of a set of revisions that they would need to do specific for each journal, and they can then make a choice. Okay, well, we'll do this set. We really don't have the time to do these. Let's focus here and go to this journal. So the goal is to eliminate the bouncing around and to make it sort of a more structured process, and hopefully give authors flexibility then in their decision-making and the revisions that they're completing. That's the goal.
Sounds good. It is optional, is that correct?
It's a pilot, and they're, I think, only accepting, I don't know what the number of papers is right now that they're accepting in the pilot, but yeah, hopefully it will go well. Yeah.
It sounded absolutely fascinating, but I'm really happy it's a pilot because there's all kinds of very interesting ramifications and those involved. So it's really kind of
Yeah. It's not. Yeah, it's definitely not a requirement. We still have the normal processes in place. It's just if
better.
people want to do it that way, they can.
Thanks. Okay, I'm going to get back to you. Sorry.
Yeah. So this is something which is very important. I think every journal tries to ensure that, but then I think it's in every scientist's responsibility also to make sure that everything, whatever comes out of your lab, is impeccable. The question is around more about rigor, reproducibility, misconduct, plagiarism. How does your journal or Cell-Press journals handle that kind of a situation? How do you first ensure that? Are there any specific tools you use to check and confirm? And then let's say if it does happen, if it's an error or intentional, how do you handle that situation?
Yeah, so this is tricky. So we have a process in place to try to eliminate some of this up front. So we have a system where all of our figures go through scans to ensure that no image manipulation has taken place. This is how we find out a lot of gels have spliced blots. A lot of gels have spliced blots. To make sure that like pictures of cells don't have background eliminated with a can from Photoshop, a spray can from Photoshop. There are a lot of that big chunks haven't been cut off of something. So we check all of that through an image scanning process. That's routine. Every paper goes through this. There can be things that slip through. So there can be image duplications. There can be some that are more egregious than others. There can be some that are accidents. There can be some that were more malicious, which hopefully doesn't happen very often. And so then it becomes a question, if we're aware of that, then it becomes a question of trying to get to the bottom of determining whether this was an accident or whether it was on purpose, whether it's the conclusions of the paper stand, whether they don't stand. Usually there's a process that involves the institution as well as the author at that point to try to, there's an investigation into what happens. It's very time consuming and it's not fun. And in the end, what happens is a determination is made, okay, does this paper need to be retracted? Is it an accidental mistake that can be corrected? And that's the ultimate decision. And it's usually takes a while to get to that point. It's very hard to determine if somebody did something accidentally or on purpose. There can be indications that might lead you to believe one way or another. But usually the process also involves the institution of the authors. In fact, it always does.
Okay, thank you. So we are almost coming to an end, but there are two last questions. And then this first question is more related to the whole scientific publishing world. And then the next one is more on career. So let's finish up with the whole, everything related to publishing. So we are in the middle of a pandemic and a lot of things are changing for everyone and everyone is feeling the ripple effects. How is your journal or the world of publishing changing? We see that a lot of preprints and, non-peer reviewed publications are coming out. So the question is more like, is that helping your journal first to fast track some of the papers who've already gone through preprint? Second, is your journal ensuring that whatever relevant to COVID-19 gets published or submitted, that gets fast tracked?
Yeah, we have, we do send all the papers to review, but after review, we do have a fast track process for publication of COVID-19 papers. We see a very large number of COVID-19 papers. So as far as how the pandemic has impacted us, I would say there's some areas in which it's impacted us more than others. It seemed at first like our submissions went way up. I think people working from home were able to get their papers out. So we had a large submission volume. We also saw a large number of COVID-19 papers, very large. A lot of them very preliminary. And so we have to monitor the bar of that area very carefully as far as what the standards that the field is expecting, making sure that what we're publishing is solid, rigorous, reproducible. at the same time as trying to get these papers out because it's a really important topic. So it's a challenging area.
The first one was, does it really help if somebody is already, in fact, not COVID, any, non-COVID situation also, if somebody has a preprint in place for some time, there's been community discussion around that paper on non-journal kind of platform.
Yeah,
Does
I wouldn't,
the journal, yeah.
Sorry.
Does the journal review it positively, favorably?
We're completely agnostic to that. So it doesn't matter to us. Yeah. If a paper's been on a preprint server or not, it doesn't matter, yeah, to us.
Okay. Anything else you were going to say? Did I cut you off? Sorry.
No, I don't think so.
Okay. So this is the last question. And so given our program, Graduate Professional Success in STEM, focuses a lot on non-academic careers also, where people are not following the faculty track. And you are in one of those careers where you're still involved in science, but it is not really a research or faculty career. Mm-hmm. So people have questions related to, how did you get into your role, what you do right now? What should trainees, PhD students, postdocs should be doing right now to be able to get in a position like yours? And do you enjoy your position or what you do right now?
So I do enjoy the position. I get to read lots of papers. And I think that's, okay, I'm not sharing anymore. Sorry.
Yeah.
Okay, great. Yeah, I enjoy it a lot. I love reading papers. So how did I get into this? I always enjoyed reading the papers the most of all of the parts of what I was doing. I loved journal clubs. I loved presenting journal clubs, finding out what was going on. When I did journal clubs, I didn't just read in my area. I picked papers from a lot of different areas to try to present, which may have driven some people crazy, I'm not sure. But that was always sort of the fun part for me. And I got to a point where I wasn't enjoying the bench work anymore. And so I felt it was time to make a change. I didn't want to do bench work. Let's see. And there was a what do I recommend people do to get into this? I would say read everything you can, read very broadly. So a lot of times we have job candidates who they think reading broadly is reading 5 papers that are tangentially related to their area. But I would say read an evolution paper, read a chemical biology paper, read a neuroscience paper, read developmental biology. Make sure you're really pushing your boundaries in terms of what you're reading, and try to see if you can read efficiently too. Try to pick out, when you do read a paper, I would say as an editor, I read a paper very differently than a scientist does. I don't dig into the figures. What I do is I try to pick out the main point. What is the main point that the authors are trying to make? What is the conceptual advance? What is the gap in knowledge? How are the authors going about asking their question and answering their question? Have they done this effectively? Does it appear to be effective to me? What's known in the field already? So sort of you have a broader perspective on a paper, and you want to make sure you can do this in a fairly short amount of time too. So if you could do this in 20 to 30 minutes, that would be ideal if you're interested in the career path.
Wow. And there's one related question, can you get into editorial board of a journal or any other related publication career after PhD, or postdoc is required, or maybe, you have to be some research project scientist position, or maybe early faculty and then get into that?
So a postdoc is not required. I would say a PhD is required. A postdoc is not required. However, most strong candidates do have some postdoctoral experience. It may not be much. It may be 1 or 2 years. But most candidates who are successful do have that. And the reason is they've just stepped outside of their graduate student comfort zone a little bit, pushed themselves, expanded their thinking in a new scientific area a little bit, and it's helped them to grow so that they're able to see things slightly more broadly. It's certainly not necessary to have faculty experience. I didn't have faculty experience. It doesn't hurt. We do have people who have been faculty members and then move into editorial. So it just depends on your particular interests at the time.
Well, thank you very much, Dr. Zearfoss. I think this was, wonderful. Thank you very much.
Okay, great. Thank you. Thank you so much for inviting me.
Sorry, go ahead. Oh,
Thank
I was asking Dr. Barron, do you have
No,
any words to
no,
say? Yeah.
this is tremendous. You had a full room and we loved every minute. This is
Oh,
incredible.
great. Thank you.
Thank you so much. This is
This,
fun.
yeah, this was fun. Thank you very much, Dr. Zearfoss, coming
Okay,
out.
you're very
And I'm
welcome.
sure your busy schedule, Zoom has made you, really difficult right now. I know all of us are busy, but still you were able to find time and come talk to us.
Well, thank you. Well, thank you for the invitation. I really appreciate it. It's been fun. And thank you for all the great questions.